All right, so we're going to pick up with muscles on, we're outside of the microscopic now, and we're going to look at how the muscle responds to being controlled by the nervous system, and then where do we get the energy to make these muscles work. So what we described as an action potential was the switching of the current on the outside to the inside of a cell that is excitable. The one that we start with is a neuron. When the neuron sends its action potential or signal down, it makes what we call a twitch on a muscle. And what's observed is this: we hit it with a stimulus, there's a brief latent period where nothing happens, and then we see a full twitch, and then it immediately relaxes. And the contraction relaxation period are the same that we talked about last time, where we saw that the contraction would be releasing the calcium that allows the troponin to open and slip our active myosin together. And then the relaxation, remember, requires ATP to break that and reopen it. So these two lines that we might notice in the lab if we were running on an experiment on muscle tissue would be the sign of, yes, I just made the contraction happen, and now the ATP is kicked in and it relaxed. But it happens so quickly that we don't actually notice that. And that's what we call a twitch. So muscle twitch is the simplest unit that we begin with. And we're going to take the twitch and expand upon that because we need multiple nervous signals to make the muscle contract and work. So here are the three phases again. The excitation contraction coupling is preparing during the latent period, but we don't see anything happen in the muscle yet. When we contract, we hit the cross bridge, which makes that tension, and the muscle will shorten. Another point that I think we mentioned the other day, but I'll just put it back in there, the tension that happens in the muscle doesn't necessarily move it. You can have a contraction where you don't move, so your muscles are tensed, but there's no actual motion visible, but it's still forming cross bridges and pulling. Those are the type of contractions we call isometric. We use isometrics in exercise and to help stabilize our body. Isometrics are going on throughout the trunk to keep us upright. So remember the relaxation period. This is when ATP kicks in. The calcium that was sitting on the troponin goes back into the sarcoplasmic reticulum. The myosin heads detach. They reset. They cock back, if you will. Tension drops back to zero, and everything goes back to its length. So they were here, and then they go right back where they were. What we need to do to control our body is make a muscle response that is graded. And we learn this very early in life. And developmentally, we begin with everything is laid into our system through pre-programmed reactions we call reflexes. So all the movements of a baby are reflexive. You stroke its palm and it squeezes. You rub its cheek and it tries to find food. We develop the ability to control our body over the first year and so of life, and that's happening up in the frontal lobe as that develops. So what will happen in time is it will leave memories and patterns of what we call the praxis of our movements, and that'll tell us from then on exactly how much control we need and what muscles we need to contract. So to control it, we have to practice and do these things over, and a good axiom to remember is that practice is how we get praxis, which is how we call motor control and planning. So our motor planning comes from practice. It's the only way to get it. Just like preparing for an exam in this class, you have to practice it. You have to do it over and over and over again. So we know just by looking at something from experience about how much we think it weighs and about how much force we need to use. When you are surprised, let's say you go to lift up a jug that you think is full and it's empty, you might find it, whoa, something light. Or the opposite, it's full of something really heavy and you think it's light and you need to adjust. That happens very, very quickly. So the two ways we grade it are by what we call frequency or strength of our stimulation. Frequency of stimulation says how often are we going to hit the muscle with neuron signals. So the quicker the signals come, the higher frequency we'll see. And then strength is a matter of recruitment. How many motor neurons are coming in to talk to the muscle, and then therefore how many muscle fibers are responding. The stronger stimulation will be more neurons to get more reaction out of the muscle, or frequency will be more signals from a single neuron, or multiple, but it's just frequency versus strength. That's how we look at it. So now we're going to take that twitch we just saw, which is here, latent contraction relaxation. But we're going to hit it again less than 10 seconds away, which we call a multiple response, and now we see a twitch starting to sum and reset. So this will make more sense when we see it on the next slide, but what we're doing here is saying I twitch it, it relaxes fully, I twitch it again, it relaxes fully. No real change, we don't really get any muscle tension out of it. So that's if we change frequency. However, if we keep hitting it, we developed what we call a wave or temporal summation. Summation means adding together. So if I increase my frequency as I go, which is, the brain knows to do this, it'll start hitting it 20 to 50 times per second. The faster it goes, the more we'll see the song. So we never go down all the way. So we start to twitch, it starts to go down, but we hit it again before it goes down. Same thing, same thing, same thing. Suddenly we get to the peak, which is the most our muscle will work. And the first phase is what we call incomplete tetany, where it's still getting summation, and then it goes to a tetany phase where it flatlines. And that's the maximum tension we can get out of a muscle. After that, the muscle will fatigue, which will bring it down and start, we'll notice that as, wow, I can't hold on anymore. You may remember the word tetany from tetanus, which is the injection you would get if you stab yourself on a rusty nail or anything that allows dirt into the body. We have a bacteria that will cause muscle contraction in the body. One of the symptoms is called lockjaw, where you lock. So tetany in the sense of here just means the muscle is no longer relaxing. To get a smooth muscle contraction, we hit it more frequently, because if we just did it like this, we'd get twitch, 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 which isn't functional. So we hit multiple twitches, and then we get the maximum response. And that works by more calcium comes out with the second hit. So the calcium that was released from the sarcoplasmic reticulum the first time is opening those connections, and then we hit it again, and it continues that before it gets a chance to relax. And that's where we see maximal tension. That's known as complete tetany, also called fuse, because the muscles lock together and hold on. However, over time, we can end up with muscle fatigue because there's only a certain amount of time we can hold our muscles that way. And that can be increased through training, of course, but it's something that is a real factor in how we work. The strength is talking now about recruitment of motor units. So if we look at it this way, looking at a fascicle, where we're going to have multiple muscle fibers or cells in there. Remember, the fascicle is the step after the muscle itself. So we have a muscle, we have multiple fascicles, and the fascicle will open into the actin myosin. So when we hit the muscle to say, I need some action out of you, I can recruit more and more and more. So they're just showing examples here by coloring them in. If I don't do anything, I have nobody recruited. But here I can recruit one. So these two little, oh, I zoomed in on it. These two little fibers get picked up. I hit it again, I get a couple more, a couple more, a couple more. This is voltage increments, meaning I'm going to hit it with more signals, but there's still, just like on the other slide, there's a point where we can't do any more. By the time we hit the 6 through 9 region here, we've recruited everybody. So with the amount of muscle fibers you have at that time, which again can be increased through exercise and training, but at this point, this is the most strength we get out of it. Or force. You can say force of contraction or strength are the same thing. So increased strength of stimulus leads to more recruitment, which leads to greater force. And if we jump back one, we see that increased frequency will lead to greater force of contraction over time as well. So we use both to decide how much effort we're going to put into a task, and our body responds to that. When all motor units have been recruited, there's no more force that we can pull out of it. And this again shows it in a different kind of loop, but it's the same image. At the first will to move, we recruit one set of motor units. Then we say, all right, we need the next group. And then finally we're going to kick in the final group and get the whole muscle going. So with most contractions we're only using a fraction of our muscle. I don't need very much muscle to manipulate these light objects, but if I wanted to lift something very heavy, I would move from here to here very quickly as I meet resistance. Those are two words that will show up. We might hit them today, but they're certainly in your future. The idea of force versus resistance. The force is what we're providing, which is the recruitment or strength, and the resistance is what we're working against. So to move an object, physics talking, we have to use our force to overcome resistance. And the biggest resistance that we deal with on this planet is gravity. So everything that we are trying to do that is up, we're always using muscle contraction against gravity. So just standing here, and you guys are sitting there, you're using muscle recruitment throughout your neck and back and abdomen to keep yourself upright. So this is where we get into the two basic types of contractions. And isometric, I mentioned earlier, meaning we use the same technique, but either the force is not strong enough to overcome the resistance. So if I tried to move the wall, if I tried to push into the wall and nothing happens, I'm still using maximal force if I push as hard as I can, but the muscle will neither shorten nor lengthen because there is no ability to move that. But more commonly, we're going to use it as isometrically, abdomen, back, neck, or just contracting and holding up my legs. Everything's there. When I start moving, I go into isotonic. So isometric by name means same length. Metric means length. Isotonic means same tone. So tone means we're still contracting, but the muscle will change in length. Now, we said that a muscle fiber, because again here we're going back to our actin myosin cross bridges, all we can do with this is shorten it, which is contractility, or relax it, which is extensibility. So we put it back or we pull it in. So we're going to work within these two parameters to say this is how we control our movement. And this is the hardest part about these two words right here. So I'm going to try to explain it in a way that I hope makes sense, but just work on those to try to separate them. First of all, note that they're both isotonic. Concentric and eccentric are both causing movement, as where isometric is not. And just remember that isometric means same length, so therefore no movement. If I do a concentric contraction, I contract. So think of con to con. And it says contraction here, try not to get confused there. Concentric means I use my muscle to shorten it, and generally I'm going to bring it up. Not always, but I'm using it to shorten, which is, as I mentioned before, all a muscle really can do. So if I tell a muscle, hit it with a neuron and say, let's go, it will shorten. So my hand, to take this pen off the chalkboard or whiteboard, I lift it concentrically with biceps brachii, and non-concentrically lifting it. So there's two options once I have it up here. I can just let go, and then I have no control, or I can lower it slowly, and I have control. So eccentric is the way down. It's not always, I guess it's pretty much always down, because we're going toward gravity slowly. So when you sit, and if you work in a long-term care or anywhere where people are old and tired, myself included, there's a lot of ploppers out there. They go to sit and they just collapse. You're missing an awesome opportunity to strengthen their leg muscles. Because if I sit eccentrically, I'm using my quads to do that. When I stand concentrically and I use them again, you'll notice the difference if you're walking uphill. You'll feel the muscles as you're coming down, like, whoa, that's a different feeling, because you're asking to do something different. The biceps do it all the time, concentric up, eccentric down. I'm eating, happening all the time. You might say, wait a minute, don't triceps brachii bring it down? Actually, they don't. You don't need them unless you're going overhead. So here, it's just biceps up, biceps down, concentric, eccentric. So an eccentric muscle contraction lengthens, and a concentric one, think of contracting or pulling in shortly. This picture, I think, puts it all together. So there's a proud papa and his baby. If I'm holding the baby here, I'm isometric. We're just snuggling and loving each other. No movement on my part, but my muscles will get tired. Or if you're carrying a baby on your hip, or carrying a bag, no movement, but your muscle will work. See, it's contracting isometric. But here, if I'm playing peekaboo or whatever we're doing here, as I bring the baby down, I don't want to drop her, so I'm bringing her down slow, and that's an eccentric contraction. But when I bring her back up to give her that kiss, concentric. So that image is one of the best I've seen over my career. I'm trying to explain what's going on here. One other thing I want you to note as we get deeper into this is that the point distally where we insert here on the radius, it's farther past the joint, meaning we've created a lever that will allow this action to lift up and down. And one of the greatest differences in someone who's physiologically designed to be a bodybuilder is a subtle difference in how far away this contraction or this insertion part is on that bone. The closer it is to the joint is it's typical, but as it goes farther out, you get more and more leverage and therefore more automatic strength built in. So somebody like me can never really get the kind of
so your average person's
but if someone's got it out half an inch farther, they have so much more leverage than they would because of that extra. It's a mechanical advantage, we call it. So we've mentioned tone, so let's get into it one more time. Tone is a description of the maximum amount of, I shouldn't even say maximum, the tension of a muscle at its resting state, when it's just kind of hanging out. And then we define people, at least in the therapy world, as being a high tone or low tone or somewhere in the middle. And I usually draw it as a kind of a dial, where somewhere here is your happy spot, and then tone that's too high or tone that's too low. This could be a temperature gauge or anything. If it's just right, this is what we call a normal tone, which, you know, normal doesn't really exist, but it's within the normal curve. This is too high, this is too low. If your tone is too high, you tend to see things lock into flexion. And these are our folks who are born with cerebral palsy or have had a brain injury, and they lock and they can't move their body because it's very high tone. If you try to help them move it, it's like pulling against steel cables. High tone is not functional because it's too tense. Low tone is called flaccidity, and this can be a result of other types of brain injury or spinal cord injury where the leg or arm is just floppy, there's no muscle generating there, and they're unable to hold their body up. Normal tone is somewhere in the middle, and we vary between this throughout the day. We tend to be a little higher tone and more upright when we're early in the day, and then as the day goes, we tend to lower tone. But the idea of it at the small level is that because of the size of the sarcomere and the Z discs, there's a point where we can't allow any more cross bridges to form. So what we're really looking at is what's the optimum length. The optimum length isn't when it's fully stretched, it's not when it's fully contracted, it's somewhere in the middle. And we've scientifically figured it out for every muscle. What's the optimum length to get the best contraction? So when you do a workout or an exercise, bringing something down slowly is going to be stronger contraction for your body, the eccentric part of it, and letting it stop just before the end and pulling it back, you'll get better leverage than if you let it go all the way. You're going to have a lot harder time bending from here than you do from here. It's just things to note. You know, if you're building muscle, that's another story, but if you're trying to help someone move, give them a chance to get halfway started. Don't try to have them stand from a deep seat or somewhere where they're already partially set. So we kind of learn where our body works best. We know this automatically, but help your clients when you're helping them move or trying to get them off the bed, raising the bed, doing things that will change that length tension relationship with their own muscles. So all I really need you to know for this page is what do we define by muscle tone, which is our optimum length of a muscle that will vary throughout the day, but it helps keep our muscles right. So the muscles are always partially tensed. And that goes back to the idea that we need the ATP running to keep the muscles relaxed. So this shows it visually at our optimal length. They're set up just like the model we used that Eric made. Says, here we are, ready to go. But if I'm contracted too much, I can't go any farther. And if I'm too stretched, I can't reach my cross bridges. So trying to get a contraction going from a full extension is harder on us than if we're partially tensed already. And there's numbers for all this. We can actually calculate the length of the sarcomeres, micrometers, and so on. I don't need you to know any of that. Just know that if we are talking about muscle tone, we want to work in the ideal. We don't want it too tight or too loose. So that's the idea of the muscle fibers. Now we have to look at how do we power these things, and this will lead us into the discussion of two different muscle fiber types, depending on what our body needs. So the first few seconds of any activity, we are drawing from muscle storage that we have stored in the muscle, and if you remember back from the old days, we are going to use glucose and oxygen as our main fuel. So a neat thing about muscles is that although we are always pulling in oxygen from our nearby blood vessel, the skeletal muscle fiber itself, here's our phospholipid bilayer on what we call the sarcolemma, and now we're inside the muscle's tissue. It's got something in it called myoglobin. So when you do AMP2, you'll learn that hemoglobin is the carrier molecule of protein that is on all of our red blood cells and allows oxygen to transport. Myo means muscle, so myoglobin is stored oxygen inside the muscle already. And that's pretty unique. But that allows us the first few seconds of activity without even having to increase the blood flow, because the muscle's already primed and ready to go. We get the inorganic phosphate transported in to set our ATP demands, so we're basically set already. We call this direct phosphorylation, which means our ATP is already primed and ready to go before we actually have to use. We don't need oxygen from the body for our first few seconds of contraction. We have a little ATP already set in there, and then that will reset it. Remember we talked about how the ADP plus P will allow us to have ATP and set it back up again. The player in here is creatinine, and you'll probably have heard of that through supplements and protein supplements that help with your muscle growth. So creatinine's ability to be used here is going to help our process. This is quick though, we only get a few seconds. So that is exhausted within the first 5 seconds. So think of the fight or flight reaction or something that made you jump and run. That first few seconds, you're gone, your body didn't even know what happened, but your muscles were ready. After that, for the next minute or so, or even less, you have to start using ATP. And we're going to do it without the extra oxygen because it hasn't arrived yet. We've already used up our myoglobin. We're going to start making immediate ATP as quick as we can without new oxygen coming in. So where do we get this ability? Well, we have glucose in our muscles already in the form of glycogen. So glycogen we've brought back to the early days, saying it was a long chain carbohydrate in which we store glucose that we've eaten in excess. We first put it in our liver, the liver converts it, and then we leave it in our muscles ready to go. So this is why we have it here. The first few seconds is preset with the activity that's ready to go, but when we hit that next phase, 6 seconds to up to a minute, we're going to take the glycogen we have, break it down to glucose, which is what we use for our ATP creation in the mitochondria, and we do it through a process called glycogenolysis. So glycogen, lysis, breaking. So we take the glycogen, take it out of the freezer, microwave it, and eat it. We give that to our mitochondria, they make the ATP, but because they're doing it anaerobically, which is the less effective way, we're only going to get a couple ATPs. If we do it aerobically, we get up to 38. Here we maybe get 2, and we're going to be left over with a byproduct. And the byproduct, pyruvate, gets transformed into something called lactic acid, which lowers the blood pH. Any acid will lower pH. But this is where we get waste product that is going to start slowing down our activity. So this is stage 2. The first immediate burst is set, but for 5 seconds, that's not a lot of activity. After that, you're going to get short-term energy. And then for the long term, now we have to go through the process of actually putting the citric acid cycle at work. We also call this the Krebs cycle, and you'll probably see this more in AP2. We mentioned it briefly as the idea of how mitochondria convert oxygen and glucose into ATP, and then give us a carbon dioxide byproduct. But this whole process, which again we'll save for another day, is a complex system known as electron transport. So what happens next is after that minute of activity, or 30 seconds if you're less in shape, after that first half a minute or minute of activity, you need more fuel because the muscles are demanding it. So we're going to pull it from the blood with the things that we have in there to use. We're going to use glucose to the maximum that we have, but we generally don't have a lot of glucose hanging around in the blood. We're going to next pick on the fatty acids and the amino acids, which are the other fuel sources we can convert. So we bring more oxygen in by increasing our breathing rate. So after that first few seconds of movement, we have to start breathing harder to work out. We're going to pull in more oxygen. The oxygen will be transported passively into here. Mitochondria take it on. They're going to arrive with some new fuel sources, such as the fatty acids and amino acids. Fatty acids look like little caterpillars to me, and these guys look like little gumdrops. But amino acids are the tiny segments that used to be part of a protein. Now we're going to use more oxygen for that first few minutes to get everything leveled off, and then if we're still continuing to actively run, we'll say we're jogging, after those first few minutes, we kind of settle off. Now the ATP process is primed and ready, we can keep pace with our demands. Now we can run indefinitely, based on our training, how long it takes for our glycogen and blood glucose to drop, how much our fluid drops through sweating, and then electrolytes that go out as well. We'll start throwing our body system off, so we'll have to eventually rehydrate. And then this whole idea is why people carb load and set their body up, or they bring those little glucose packs and just suck them as they're running. This will help maintain that. So from there, as you know, with marathon runners, we can continue to exercise for quite a long time once we set this pace and continue to refuel it. So again, the summary here, at this last phase of long term, we need oxygen. It's therefore called aerobic. Remember, air and oxygen go together. Now the ATP will not come out as quickly as it did, but we'll get more per unit. So we'll get up to 38 this time instead of the 2. So we get these 2 very quickly by hitting the glycogenolysis and doing it without oxygen. But for your money, you're better off getting it here, taking a little longer, but getting 38 out of each transaction. And then again, we're going to use the pyruvate, which is what we broke down in the glycolysis, fatty acids and amino acids, and that will lead us to all the waste products that we get out of the kidney when we're going to deal with this later. So the NH2, which will become a waste product, creatinine from the creatine will be a waste product, and that will all help us clean that out of our system. But this is how they work. This image, I think, again, if you like images, really sets the stage. We have 3 runners on the track. This first runner, who was just doing a 50-meter sprint. In that first 5 seconds, they finished their sprint. That's a fast run. They finished, woohoo! All they used to do that with the ATP they had in there and the immediate phosphate transfer, they're set. The next runner is going to continue on longer, but only going to go about 50 to 60 seconds. Within a minute, let's say they finished the 400 meter, they've used glycolysis to get there. They broke down the glycogen they had, but again, they don't have to increase their breathing. This person who's doing the 1500, going a few laps around, taking 5 or 6 minutes, is going to move to what we call aerobic cellular respiration, and that is how they manage to do that. So your training and what you're trying to accomplish depends on how your muscles are going to react. With that in mind, we will then realize that our body will use different types of muscles for different activities, and it will learn by training what you want it to do. So just as we used practice to teach our body how to use its motor patterns, we now use the conversion of 2 different types of muscle fibers to set our body up for what we are planning to do with it, and that's what training is. Again, genetically, there are certain advantages people have. There are different physiologies that are more suited to different kinds of activities than others. You'll tend to see sprinters being the shorter, squat fellows and ladies, and then the taller, lanky folks tend to be better at how they do long distance, but it varies. But that, you look at different athletes and their size and shape and their proclivities, it kind of sets up physiologically, maybe they're designed better as a unit. So when we hit fatigue, which is when we hit that edge of, and there is a literal runner's high that comes with that. If you are a long-term runner and you start hitting this phase, your body will increase the secretion of dopamine, which will make you feel elated and allow you to push on. But when we hit to the point where we just can't go any farther, these are the things that run out. Glycogen's used up, so ATP synthesis goes down. Without ATP being readily available, the sodium-potassium pumps will start to fail. The sodium-potassium pump will be brought up very soon as we get into the neuro unit, but that's I hinted at it before, but that's the charge differential that we get between the membrane, which is ATP driven, that says any sodium that leaks in, I'm going to kick it out. Any potassium that leaks out, I'm going to pull it back in. And by doing that, actively running that pump constantly, we have excitability, which we call active, well, it will lead to an action potential, but right now we call it a resting potential. So that will start to fail. The lactic acid we secreted back on phase 2 will lower the pH of the sarcoplasm, and anytime we screw around with pH, we're going to potentially denature an enzyme, but at least it'll inhibit it at this stage. And then as we get more potassium extracellularly, once again the membrane potential excitability will throw off. And then the other player, who's the one who's talking to it in the first place from your brain saying, please run for me, I need you to run. Remember, he's filled with acetylcholine at the end. The acetylcholine will eventually use up, and that again will stop the signal. So building endurance, building muscle, we are training our body on a regular basis that this is what we need you to do. The body's, I think you said low-key lazy, but the body's inherently works at its lowest level of activity because it takes a lot of work to make us do everything. So if we're not asking it to do anything, it'll say, great, I don't need to make all this stuff. But if you are regularly getting up and pushing your body to use all these activities, we will gain over time all the right things to meet our needs. To get back to the way we were, which is our pre-resting state, we have to really bring in some oxygen and remove the carbon dioxide. So to get back to where we were, we call this catching your breath. We're going to have to replenish all the oxygen reserves that we used up. So when you do A&P2, we'll talk about how hemoglobin carries it throughout the body. And an interesting thing to remember is that hemoglobin will be very guarded with its oxygen release, and it will rarely, if ever, give up all the oxygen it has. It will give you some, and then it'll keep adding more because it doesn't want to run out. So you've got that going, but you immediately used up everything you had in your muscles. Your myoglobins are depleted pretty quickly, so we have to get those replenished. The lactic acid you're building up, which is going to lead to the muscle soreness, has to be broken down now, and that'll get that out of your system. Your glycogen has to be replenished in your next meal, and we have to put the ATP and creatine phosphate back to the way it was. So all these steps, you have to get more breathing going, and that's, you think of it as repaying back the debt you just used. And you'll notice that if you've done it yourself or watched others, when you get to the end of a race, we often breathe in this position, which uses your chest and neck muscles and shoulder muscles to lift your ribs higher to get oxygen in quicker. So typical breathing, we breathe out through our diaphragm and push the belly out, but when you force inhale, like at the end of a race, you often have to recruit the rest of these muscles to help you breathe there. And that's again repaying the debt. So here's the type of muscles that we, and I unfortunately have to go the way I like to understand it, and maybe it'll make you remember it or make you grossed out, is the types of meat we eat on like a chicken or a turkey, white meat and dark meat. So I will name it that way, but learn them as slow twitch, fast twitch, but if that correlation helps you, you'll always think of me when you have a meal. So we have fibers in a muscle that we know, and they're doing their thing, but they're going to have a little subtle difference in how they're arranged. And that will be, again, dependent partially on genetics and partially on, mostly on how we ask our body to act. So the two types that we define them as is slow twitch or fast twitch. A fast twitch fiber will fall under the white meat category, but the slow twitch would be the dark meat. The way you can help remember that is that the capillaries are much thicker here, which makes it much more full of blood, which makes it appear darker. These guys are resistant to fatigue. These guys have much more set up with mitochondria, myoglobin, and capillary input, and they are adapted for aerobic. They are very close to the oxygen supply, and we need this to hold on. Our back muscles, our leg muscle, the soleus, all these back here, all these guys are going to help us hold our body up, and they're not asked to do a lot of things. But on top of soleus is a fast muscle, gastric, who will spring into action. And we have other fast muscles that we're asked to do for power. On the bird, a fast twitch muscle in the breast is a bird that doesn't fly very far. So the chicken or the turkey that squawks about, takes a short flight and lands immediately, and we breed them this way, they're going to have the lighter meat in the chest or pectoral region, which is what we see as a fast twitch. They're rich in enzymes, and they're ready to do that second phase of glycogen and lactic acid. They release the calcium very, very quickly, but they're low on blood supply, myoglobin, and capillaries. So they're designed for this phase of the run. Here, or in here, both of these. But the slow twitch muscles are designed for the marathon. So when we look at it that way, that makes sense why the game birds, like a goose or a duck, if you've ever eaten those, they are all dark meat because they fly days, miles. Where our domestic birds that we raise for food, we design them this way over the years. So that's what we mean by fast versus slow twitch. So when you are training to be either a lifter who needs fast twitch to do explosive power, or a sprinter, or jumping and leaping, or you're training to do a long distance run where you need endurance or maintain your body, you're going to change the physiology of your muscles on the inside by bringing in more blood or releasing the blood and converting it to fast twitch. We generally, as I mentioned about the size of the different athletes, we generally don't see an endurance athlete with jacked muscles. They're going to tend to be leaner, and they're using a very different mechanism, the oxygen mechanism, to give them much more power over time. Other things that we know about oxygen intake, or uptake I should say, depends to be on your size, and because males tend to be a little higher and larger, they tend to get better oxygen uptake. Your best oxygen uptake for your life, we all probably all peaked out already, when you're around 20. But if you're a trained athlete, you can maintain that throughout your life. Humans pretty much go, whoop, and then the slow decline. But we don't need that, we can keep going. But again, if you're going into long-term care or working with people who are debilitated, you're gonna see a lot of them way past their highest peak, and just walking from here to the door will fatigue them. They'll be fighting for that breath the whole way. And again, we in therapy are trying to train them and get them back into endurance and get them strong again, but it's a long road. That's not at all like it was when you were young. What I do notice though in my own practice is that if someone was trained when they were younger and has a memory of being in shape, it's easier to get them back there than if they were sedentary their whole life. That's a tough lift. But if they're, they say the word muscle memory, which is not, the muscles don't remember, but the brain remembers and tells the muscles, remember you used to do this, and it brings us back. The other thing that will affect our endurance is what you have in your diet. So fatty acids, amino acids, and glucose are the fuels. We need those. We need the amino acids to build the muscle, and the fatty acids will be another form of fuel over time, where glucose is the immediate fuel source. And then carb loading is, as I mentioned, a way to pack the body with glycogen ahead of an activity and you're ready to go. The strength of the contraction, as we said, is dependent on how often we hit it with a signal and how many motor fibers we recruit. So if we're building a body to be powerful and build muscle, we're going to train it a little differently than if we're training for endurance. And we call that, your aerobic exercise versus your strengthening exercises. So how many motor units and recruitment will change with your activities. You get one axon per neuron. So this neuron that came from the spinal cord that's talking to this muscle, that's all he's got. But if you ask him to do more over time, he will grow branches and build a better connection and more acetylcholine with more receptors to say, this is something I'm going to do all the time, so I might as well set up. And we do this in our brain when we focus on learning something. We build better connections between neurons that we need over and over again. So this is what we call neuroplasticity, and it happens at the nervous system level in the brain, and at the neuromuscular level with how we control our body. So someone who learns to play an instrument has developed a different set of fine motor control through that left hand than they might if they never played a fretting instrument. And then this side of the brain, the control of that, will have more diverse neuron pathways and networks than you would if you didn't do that. So learning multiple things at an early age, when we have the most plasticity, really sets us up for a lifetime of enrichment, which is why you see a lot of attention on early childhood education and exposing to as many things as you can. You can learn languages and speak like a native when you're an infant and a little baby, but later on you'll always have a little accent because the brain works that way. It kind of sets in its ways over time. But anyway, back to muscles. The size of the motor unit and recruitment is going to affect it. How often, as we mentioned, we stimulate it, and then this length of the muscle at the start. So you can get a better power out of setting your body at the best. And you watch power lifters and you see how they set themselves a certain way before they begin. Over time, with resistance training, which is weightlifting, we're going to ask the cells to grow because they're going to make more myofilaments. So actin and myosin, which were proteins, so they came from the proteins you're loading, usually those giant tubs of amino acids that they're just eating and putting in all their drinks. A lot of us do it now. We're going to put it in here and we're going to build this muscle over time by saying, I need more of these. But if you add the amino acids, you don't ask it to do anything else, now you just have extra amino acids in your body and it's waste product. So you have to combine it with, I have the demand, and then I'm going to supply it with the building blocks it needs to get bigger. So that's called hypertrophy. The opposite is atrophy, without growth. Atrophy is what happens when we leave it alone. And it happens quite quickly the older we get. So your clients in your future practice who may have had a fall or whatever happened to them, they maybe been in bed for a week, they've atrophied already. They're not, even 2 to 3 days they atrophy. And they're not as strong as they were, and their respiratory endurance is low, so we have a long road back for most of our people we work with. So we're building more myofilaments there, and then with endurance, we don't have to build more myofilaments, but what we have to do is make more mitochondria and the enzymes, the glycogen storage, and make more capillaries. So there's a subtle difference between how the muscles are going to be, and we can switch this back and forth. If you have been resistance training for a while and then you start to go into endurance, the muscles will start to shift and say, okay, we're going to add more capillaries in, and we're going to start shrinking these down to make better use of what we have. And then we'll just throw a couple of diseases in there. These are never things I test you on, but you may have heard of them. Muscular dystrophy, and this tends to affect males, and it tends to be fatal early in life. Is a genetic disorder. The muscles degenerate and weaken. Too many calcium ions get in and destroy the proteins, and then the dead muscle cells are actually replaced with fat and connective tissue, so they no longer can contract. And a common fallacy, I guess, or a myth, is that if you don't exercise, your muscle will turn to fat. That does not happen outside of a disease process. What happens, however, is that if you continue to eat the calories you did, and you're no longer asking your muscles to use that much energy, fat will grow on top of your muscles, superficial, and you'll look heavier than you did when you were fit. So people will change from bulk muscle to bulk belly because they've given up on the part that burned the energy and just keep adding it, and then the fat tissue is like, hey, I want to use this, I'll save it for later. Maybe someday he'll want to work out again. Then ALS, which Lou Gehrig famously had in the, I can say 20th century now, the mid-20th century, was a ball player who was struck with this horrible disease called amyotrophic, which means without muscle growth. Lateral, which is a specific part of the spinal cord, sclerosis is hardening, is a change in the spinal cord that cuts down the motor neuron activity to talk to a muscle, which would lead to loss of motion, usually distally first, fingers and toes, and then paralysis up, right up to here. You keep your ability to think.
It's probably the worst disease.
Yeah, but you can't speak and breathe eventually, and you have your brain, you're fully alert, but everything shuts you down. It's a tough one. We have an ALS center right at St. Peter's. It was regional throughout the whole upstate New York and beyond, and I've worked there as well. And again, like you said, it's a horrible
My
fate.
father passed away.
Yeah, it depends how long, there's detractors that will say how long you live, but it is a death sentence. So that's that. We'll end on a bummer there. So let me open up the one on the nervous system, part one. Any questions so far on what we need to know about muscle? This third part, I think, is the easiest part, because you're just memorizing a couple different scenarios, whereas before we were going into submicroscopic level, trying to figure out what goes on. This will be very familiar because we already did this. You recognize the end bulb of an axon. You recognize mitochondria sitting in there. These little fibers are just part of the cell structure, but all of these guys here are your vacuoles or vesicles that are holding a neurotransmitter. A neurotransmitter is another way to describe a specific type of hormone, and these are always used to communicate. So this gap here, which is called a synapse, is the space between neuron end number one. In this case, it's going to be neuron number two, but as we just saw, it could also be a muscle. The only other thing you connect to, besides another neuron or a muscle, is the gland. And that's AMP2 as well, but if we put an endocrine gland here, this signal will tell it secrete your substance. If I put a muscle here, this signal will tell it squeeze. And if I put another neuron here, the signal will say either send the message ahead or stop sending your message. And those are called excitatory or inhibitory. So it's just an on-off switch. So that is a simple way to look at it. It's just communication between two structures. But we have trillions of these, and we have beyond fathomable ways to communicate and build networks. We're like an entire internet up here, and we have an awful lot going on at all times. So we'll give you the basics, and then we'll try to move into, we'll leave this off before we get into the heavy stuff for this exam, but for the last part of the unit in this course, we're going to deal with the brain and all that stuff that gets super complex. So the first step is to use the definitions that we have come up with to describe what the different units of the nervous system are, and this again is just a definition for us to start with. We're going to describe something as either central or peripheral. Central meaning in the center, peripheral meaning on the outside. The way to define it is if it's protected by bone, which would be our old friend the skull, the cranium, or the vertebral column, that is called central. So the central is literally the brain itself, behind the skull, and the spinal cord, which travels right down to about there, which is inside the vertebrae. It stops here in the beginning of the L region. The lumbar one is about where it ends, and then from there it's just nerve fibers. So another way to think of the word nerve is it's always peripheral. Anything that's a nerve is in the peripheral system. So we have nerves that come out to our eyes and ears and face, those are peripheral. We have nerves that go all the way out to every other part of our body, those are peripheral. So everywhere they are is the peripheral system, which again is not inside the skull vertebral column.
I just have a question for you.
Yes.
Recording. Do you want to pause it and redo that?
Do I?
Or are you keeping it?
I'll just put it together and I'll edit.
Okay.
Thank you. Yeah, I'm probably gonna have a giant video, but we'll do our best. So the other thing to note is that as we break down how the neuron works, we're gonna have an axon and we're gonna have a cell body. And off the cell body will be dendrites. These cell bodies are super fragile. The axons can be replaced. They're long tubes that will communicate as far down. So each of these nerves is just bundles of axons. So you can see a single nerve could begin your spinal cord and travel the length of your arm, or begin in your low back and travel the length of your legs. So you can get like a 3-4 foot long cell, which are even longer than the muscle cells we talked about. But the only part of that cell that is that long is the axon part. The cell body, or soma, is protected elsewhere. If it's central, it's inside the brain or the spinal cord in what we call the gray matter. We'll get to that later. But if it's peripheral, the cell bodies show up in these things called ganglia. So ganglia, by definition, is a cluster of cell bodies, like watching these guys hang out together. And this makes a ganglion, but ganglion are only found in the PNS, peripheral nervous system. In the central nervous system, if we have a ganglia, a cluster of neurons, we call it a nucleus, which is the third time we've used the word nucleus, but it always means the same thing, a central cluster. So we use nucleus to describe the inside of an atom, we use nucleus to describe the brain of a cell, and now later we'll use nucleus to describe specific regions in the brain that are clustered together. But out in peripheral, we'll always call it ganglia. Ganglion is one, ganglia or two or more. You'll notice the ganglia are set up in very specific regions. These are called paravertebral, around the vertebrae. We have a big one right here called celiac, which we also call the solar plexus. He's not here on this picture, but it's a huge second brain right here. And then two more, superior and inferior mesenterics, that go here and here. They run this whole system here. And then the other ganglia are always going to be as protected as we can get inside the spinal cord. So we'll name those later, but just know that if you see the word nerve or ganglia, you know you're talking peripheral. And they're cell body or axon. Ganglia are the body, nerves are the axons in bundles. And if it's central, it'll still have axons, but they're going to be a lot closer together, and they're going to be designed with the word nucleus as a cluster of them in the cell bodies. This I drew way in the beginning, and I always draw it this way, but it's the same. I'm going to come up to the brain and down from the brain. And the brain, I'll just write CNS now, because that is the brain, which includes, of course, the spinal cord and your gray matter and white matter up here. We're always going to come in. I got a brain tumor here. I'm gonna fix that poor guy. He's not looking good. There he goes. I'm trying to make it a little more brain-like. We're always going to come in through what we call an afferent pathway. And we always go out through an efferent pathway. And this is the same if we're coming in directly to the spinal cord and out, or the brain itself and out. So if we come into the spinal cord, all we do is go up to the brain and back down again. There's certain words that I want you to remember for the rest of the semester and beyond that will help you make sense of what we're talking about. So I'll list them, here, move this, I can list them in columns. When we come in, we're receiving information from our environment, and it can be the external or internal environment, doesn't matter. We're always receiving information through dendrites. Dendrites are sensory feelers. So these little feelers are detecting things inside our body and outside of our body, and those things are always called stimuli. So a stimulus is simply something that has changed. If everything's the same, the body is key to detect any difference, and when something changes, it notices it. Whether we notice it or not is up to the brain. The brain notices everything. We assume that someone in a coma knows everything, but their awareness, who they are, their consciousness is offline, so we don't know. So when you're sleeping, you're not aware of what's around you, but your brain is. Your brain's listening, your brain's processing, it's always busy. So stimuli are always being processed in here. This is how they're processed. They receive the stimulus. There's something called a receptor. I kind of set that up for you. A receptor is the business end of a dendrite. Something changes, the dendrite notices it, and the only thing it can do is send a signal. The signal is that action potential that we talked about that is flipping the positive and negative. And by doing that, it goes up to the brain, or the spinal cord, then the brain. So the words that we'll have over here are afferent, ascending, you'll see that word a lot, and sensory. And then other words that make sense in here would be up and in. So they go up or in to get to that information. And we're going to lead it up to the brain, but the other word that will always go with this is dorsal. Because information enters the brain through the back and enters the spinal cord through the back. So the other word for dorsal is posterior. So anything that fits on this side of the column is incoming sensory information. That's this side. Or the way they did it, the top. The brain, central nervous system, including spinal cord, integrates and interprets. So just put those words up here. Integrate and interpret. The subtle difference there is, I'm going to, no, it's okay. I can't talk right at the same time anymore. Is I'm going to collect every sensory information that comes in, telling me everything that's going on around me. The temperature, the light, the movement around me, the thoughts that I'm worrying about, the clothing on my body, everything's coming in and getting classified. What we call or talk about with attention deficit is that we can't integrate it well. It's just too much. It's flooding in and we're like, ooh, what about that? Oh, look at this. Oh, and that's frustrating for learning because while it makes curiosity and it makes you really interested in life around you, you have a harder time focusing, which will put the information down into permanent storage. So a lot of it's just whizzing by. So the difference between someone who is attention deficit would say that I'm gathering all this information, but I don't know what to do with it. So that's something to be trained or modified with chemicals. Interpreting it is saying, is this something I need to pay attention to or not? And this is usually where we're going to fall apart at that level. Is we're interpreting it as a threat. Now I've changed my whole demeanor on, oh, I got to run or I got to fight. If I'm interpreting it as something that needs my attention, oh, a butterfly. Now I've changed that as well. So this job of the central nervous system is ongoing, and then our consciousness exists somewhere in here at this stage when we're alerted to it. So how much you're alerted to your environment is again that kind of idea of how your attention works for you. But we do know that you can't really learn without sustained attention. So that's where we run into it all the time in education, helping children and adults process how their brain works so that they can be functional and get what they want out of life. Efferent is the way out. So we're going to use all the opposite words over here. So again, I'll say efferent. The opposite of ascending is descending. The opposite of sensory is motor. So this is what we're going to end up down here. And then the motor is called an effector. So even though I have this word outside, I should include it in my box. Receptor goes in there, effector will go over here. Up, in, down, and out. Dorsal, what's in front of that? Ventral. And the opposite of posterior is anterior. So all of these words will help you through the rest of the nervous system. Like when you hear them in different contexts, you'll say, oh, well, if it's efferent, descending, motor, down, out, ventral, anterior, it has to be motor, which means it's leaving the brain, going to an effector. We only have 3 options. We can talk to a smooth muscle, which is involuntary. We can talk to a cardiac muscle, which is involuntary. I guess we'd have 4 options because those are 2 different things, so we can do a 4th option. We can talk to a gland, which is involuntary, or the only thing we can actually control is a skeletal muscle, which you are learning in labs now. And you'll start naming them, but know that that is the only voluntary effect we can make on our environment. These guys are involuntary. Glands, cardiac muscles, and smooth muscles are running on their own. It doesn't mean that we're not in control of them. Our brain controls it, but your consciousness is not allowed access. These are the programs that are running behind the scene that they don't want you messing with. because we would make a mess of ourselves. So skeletal muscle we can control. So the way this loop works is, sensory information comes in, I see a glass of water, I'm thirsty. The brain detects that. I know what that is, that's water, I want that. Motor output says, I want to make my muscle go get it. And then I do that, and then I close the loop, and I'm happy. Then the next sensory input would be, I swallowed, and then the motor input would be, I'm going to use my muscles here, and that's what we do all day, all the time. So if you see it as this or that, doesn't matter the order. I like it as a up and down, because here's where the brain is in the body, but that they show it this way, it's the same thing. So here's our choices. Now, we're going to break it down one further by saying that when I say voluntary or involuntary, involuntary means autonomic, which sounds like automatics. That's the best way to remember it. Autonomic is taking care of itself. Voluntary is called somatic, because soma means body. So that means we can control it. So on the output, you'll notice that as I send messages out to the effectors, which are smooth muscle, cardiac muscle, glands, or skeletal muscle, 3 of them are autonomic. Cardiac muscle, smooth muscle, and glands. I don't have any say over that. Well, you do, in a roundabout way. If you get a sensation that's going to make you nervous or upset, all these things will react. And many times the sensation that we get is thoughts, not actually anything that's happening. Dread, anxiety, fear, nothing's really happened, but we're thinking. So thoughts actually control this, even if there is no sensory input except your own worries. And that's again where mental health and anxiety come in, because you can stop that by changing your thoughts, which is the basis of cognitive rehabilitation through cognitive behavioral therapy, and then we can slow down all this and get it back to where it should be, but that's for later. But the only motor nervous system output that we have control over is the somatic one, which is skeletal muscle. So that's how I broke them down here. Now similarly, we have 2 different types of input that we can receive. Same thing, somatic or autonomic. So what somatic means in this case are things that we are made aware of that we can feel. So somatic sensory comes from what we call the senses. And you notice after the 5 senses they say plus proprioception, because there's really 6 senses. I would say 7, but we're not going to get crazy here. But the 6th sense is not seen dead people, it's called proprioception. So the 5 senses being touch, which is everywhere in your body. Everything has the ability to detect touch. There's no part of your body you don't want to know that you're being touched. Sight, you only get that through the eye, which is the retina in the back. Hearing, which you get through the inside of your ear. You also get vestibular on the inside of your ear, which is what I would call another sense, the sense of where you are. Is your head moving or not? That's a very important sense you get in your ear. Taste, which you only get on the tongue and around the inside of the mouth with what we call taste buds. And smell, which we get on the inside of the nose through little dangling smell receptors. Each of them has this very specific nerve in the brain, which although they're nerves, instead of the spinal cord, they go right to the brain, so we call them cranial nerves. You'll learn all those before the semester's out. But they are telling the brain, I've detected taste, smell, scent, all that stuff. Smell and scent, the same thing. I've detected all these things. Proprioceptors are inside all of our muscles and joints, and they tell us what our body is doing at any given time. Am I standing? Am I sitting? Do I have contractions? Am I relaxing? So the ones that we know as the 5 senses, we don't include proprioception, and I would add vestibular as well, which the book didn't, but you gotta know where you are. Vestibular is specifically the head, where proprioceptive is the rest of the body. And then visceral. So it's still autonomic, but we use the word visceral as opposed to somatic, because visceral means your guts. Somatic means your body. So yes, your body includes your guts, but here we're referring to skeletal muscle, here we're referring to all the rest of the organs. So what does that mean? Saying that they're not in the same category as the somatic. That says that your brain is well aware of what's going on in every tube of your body, but you don't need to know that. You don't want to know that. You don't want to know, how things are moving or whether the blood, just too much information. You need to know if something's wrong. So if there's pain, if you get cramped or you have a blockage, you'll be made aware. So the only real sensation we get out of this is pressure and pain. So if there's pressure and pain in one of your tubes, you will be made aware and you'll have to do something about it. Go to the ER or go to the bathroom. Usually it's 2 options. Or just, if you're a man, you'll ignore it. So we're afraid. We're very afraid. We go, no, I'm fine, until we drop dead one day. And this sets it all up in a one big picture. So again, here's our brain and spinal cord, and it's all of its glory. We're showing information going in and going out. So the reds are always going to be output in the way they draw it, which are my right side of the board. The blues, as they're showing it, are the input, which is always going to be sensory, which is my left side of the board. So there's that word, afferent. Now it's easy to do the afferent side, because all we have is we're going to bring information in either from a visceral or somatic. Very easy on that side. It's either going to come from a gut organ, like the stomach, or something like my skin, where I have all kinds of little, remember we named a bunch of them in the skin? The merkles and the other fibers, push on them. We need a lot of hair, we're made aware of movement. This side is a little more complicated, because on the efferent side, or the motor side, we immediately have to decide, am I going to control a skeletal muscle, or am I going to control something else? If I control a skeletal muscle, that's easy. I voluntarily go to somatic nervous system, skeletal muscle, and done. But every other gut, every other part of your body, with one exception, is going to have what we call dual innervation. So we take a heart, we take a bladder, we take all the intestines, we're going to hit them twice. We're going to give them one fiber that's called sympathetic and another one that's called parasympathetic. And we're going to take our turn depending on what the needs of the body are. So the cute ways to remember them are sympathetic is fight or flight, parasympathetic is rest and digest. Where their names come from is how far up they are on the spinal cord. So sympathetic is in the middle, and para, which means around, is up here and down here. So parasympathetic signals come from the base of your brain and the bottom of your spine, where sympathetic come from the middle. That's where the name came from, just to clarify. But they're all motor, and they have a choice of either increasing the activity for fight or flight, or bringing it back down for rest and digest. So it depends on the organ. If I take a heart, that's very easy. In a fight or flight situation, I'm going to make that heart beat fast, because I need the blood to get to the muscle to get the oxygen to the muscle so the muscle can run. Which again, after the 5 seconds, I got to keep running. So you can run from a bear for 5 seconds, but that's not enough. You got to keep going. So you need the heart and the lungs and everything else to speed up. What you turn off in that case is the guts. I don't want to be wasting all my energy on digesting food. So I shut down all of this. So sympathetic effect on your intestinal tract is inhibitory, shut it down. When I jump back here to show you this scenario, you're going to notice, is that where I want to go? Yeah, here. Notice that all these little knobs that I said were ganglia, they're all arranged in the thoracolumbar region, between the chest and the low back. Thoracolumbar is another name for the sympathetic system. The name for this, parasympathetic, is craniosacral. So if you heard of the craniosacral system, that's calming your body, putting you back down. And the biggest player in this is the vagus nerve. It comes out of your cranial nerves and says all this, get back to work, digest slowly, take it easy, calm down. At the middle, I look at these strings of fireworks. They're all lined up, and I light one, and the whole thing goes pop, 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 and your body is in rage, or run, fight or flight. So that's how they're arranged. We'll name all that later. In lab, you'll spend a lot of time on those, naming the different ganglia and so on, but just know that rest and digest is where we want to be all day, every day. Fight or flight is only for emergencies, but in our modern world, we find other ways to use it, and we set ourselves up for all kinds of stress events that aren't necessarily life-threatening, but we interpret them as such. It's almost like this system, it kept us alive through the millennia as we were growing into an upright creature that has thoughts and does things instead of just an animal. Most animals run in, wild animals run in sympathetic all the time because they're always afraid of getting killed. We got through that, but it's almost like that system is so powerful that we need a use for it now. So we find ways to make ourselves upset, and it's not helpful. So I'll leave you with these thoughts. This is what makes a neuron different than all other cells of the body. The first two are the same that we saw in functions of the muscle cell. They're excitable. So the excitable cells of our body are neurons and muscles. Excitable means that if you hit it with a stimulus, it will secrete something at the end, or it'll shrink, or it'll do something. It'll respond in some way, which again is this loop. But the way the nervous system responds is continuing the signal along, and then usually secreting something at its end. But what we need to know by the definition is that we change the membrane potential when we're excited, meaning we open the gate that changes the sodium-potassium relationship, and that changes how our polarity is, which will take us back to the action potential and save that for next time. Conductivity means if I do something here, I can send a signal to the end. So important. Similarly in the muscle, if I hit here, I'm going to use my T tubules to send the message all the way through, and then the whole muscle hears about it. So this scenario with our neuron, I use the axon to talk to the next structure. Some axons are so short, we're like right next to each other, and others, as I said, are like 4 feet long and go all the way down to the little toes. They have the ability to secrete the neurotransmitter at the end, which we call the axon end bulb. We fill it with vesicles of what we made. Acetylcholine is the one we've learned so far, but there's others. I mentioned dopamine, that's one. That'll make you feel high or happy. And that will tell the next thing to do something. We use exocytosis to get it out. Whenever we say exocytosis, we know we have to use ATP. So these, just like neurons and muscles, neurons are just like muscles, I should say, are always humming with energy. They're always using ATP, they're always busy. They need to maintain that high level, and they're the biggest users of energy in our body. When they hit the target cell, the two biggest things they'll do is either stimulate it, which is what a muscle does, or inhibit it, which says stop doing what you're doing. They live very long. The neurons we have can last you your whole life. But the downside of that is that they don't do mitosis. So if a neuron dies, that's it. So once you're done with fetal development, the neurons you have are the neurons you have. So you might say, well, how does my brain grow or change? It's the connections. It's how many of these I make, it's how much neurotransmitter I release, and it's how I network them together. I bind them together in ways that will make pathways that will be permanent for me. And then to change those pathways, which we usually call habits, is very difficult. You have to change your brain chemistry to get there. So we literally change people's brains when we're working with them in cognitive rehab therapy or anything that's helping them strengthen their body or change. So we'll call it a day here, and we'll pick up on next week.